In the dimly lit laboratories of leading scientific institutions, a new frontier of human understanding was unveiled. In a groundbreaking advance, scientists had taken a daring leap into the world of synthetic human embryos, pushing the boundaries of science and ethics. Using the power of stem cells, these visionary researchers sidestepped the traditional need for eggs and sperm to create something truly extraordinary. These synthetic human embryos resembled those in the earliest stages of development, offering a tantalizing glimpse into the intricate dance of life's beginnings. However, with this bold step came serious ethical and legal quandaries. The lab-grown entities existed in a realm outside the bounds of current legislation, not just in the UK but in most countries around the world. They were, as yet, devoid of a beating heart or the rudiments of a brain, but they held within them the potential to form the placenta, yolk sac, and the embryo itself. Professor Magdalena Zernicka-Getz, a luminary from the University of Cambridge and the California Institute of Technology, unveiled this pioneering work at a prestigious gathering in Boston. She explained how human embryo-like models could be crafted through the reprogramming of embryonic stem cells, unlocking the mysteries of that elusive black box period of development. This black box, a period during which the law restricts the cultivation of embryos in the lab to a mere 14 days, has always been an obstacle for scientists eager to understand early development. Until now, they had to rely on pregnancy scans and donated embryos for their research. But this revolutionary work promised to change everything. In previous experiments, stem cells from mice had self-assembled into early embryo-like structures with developing organs. It was a remarkable achievement, but the real race was on to replicate these early stages in humans. Several teams had managed to do so, mimicking the very beginnings of human life. The specifics of this latest work, however, were shrouded in anticipation. Zernicka-Getz hinted at her team's success in cultivating the synthetic embryos just beyond the 14-day milestone. These structures, each derived from a single embryonic stem cell, reached a stage known as gastrulation. Here, the embryo underwent a transformation, differentiating into distinct cell lines and laying the foundation for the body's basic structure. At this stage, there was no heartbeat, no developed gut, and certainly no brains, but these tiny models revealed the presence of primordial cells, the building blocks of future egg and sperm cells. It was a revelation that left the scientific community in awe. Yet, as the science surged forward, the laws lagged behind. Scientists hurriedly began drafting voluntary guidelines to govern their work with synthetic embryos. The concern was real, for these creations were rapidly becoming more akin to natural embryos, and the question of how to treat them weighed heavily on the minds of researchers and lawmakers alike. And then there was the ultimate enigma: could these synthetic embryos, in theory, evolve into living beings? The mouse embryos appeared almost identical to their natural counterparts, but failed to develop when implanted in female mice. Similarly, monkey embryos created from synthetic cells showed initial signs of life, but could not sustain beyond a few days. As scientists grappled with the tantalizing possibility of life emerging from their creations, one burning question remained unanswered. Was this limitation a technical hurdle or a fundamental biological barrier? The story of synthetic human embryos was still being written, a narrative of scientific ambition and ethical contemplation. With each new discovery, the line between possibility and ethics grew thinner. The need for clear legislation had never been more pressing, as humanity ventured into uncharted territory, pushing the boundaries of what was once deemed impossible.